Hello friends, welcome to my channel Clinical Biochemistry by Dr. pkprabhakar. Today we are going to see one of the important basic topic where we will see what is the significance of DNA supercoiling. Whenever we are going to study DNA structures, their packaging, so that time we are talking about the nucleosome models. So supercoiling is there, nucleosome packaging is there, but what is the exact significance, how it is important for the cells? So that 5 important points how supercoiling helps our cells and what is the reason behind that one, that we will understand. So first basically we should see what is the size of our DNA. If you will see the total base pair in the human genome is roughly 3.3 multiplied by 10 to the power 9 base pair, and the total weight, if you are going to see for every base pair on an average weight is 650 daltons, so it will be equal to 2.15 multiplied 10 to the power 12 dalton. 1 dalton is equal to 1.67 multiplied by 10 to the power 24 grams. So the human weight will be, if you are going to multiply this one by 1.67 to the power 10 to the power 24, this will be equal to this much, and this is means it will be roughly 12 grams. So if you are going to collect all these DNA from a human cells and going to weight it, it will come around 10 to 12 gram. Now this is the weight, but if you will see the lengthwise, so the total, if all the DNA of a human cell will be placed end to end, so it will be roughly 6 feet long from a single cell. But if you will take all the DNA from all the cells of a average human being, and it will be that big long, so that you can go from here up to the sun more than 300 times. So this is that much big long. So because that much big long, and our cells, you know that it will be in the nanoscales or the micro scales, so this much big long or 2 meters long DNA will not be accommodated into the cells. So how this DNA is going to be accommodated into the cells which is at the micro level? For that one, normally we are going to have DNA supercoiling, or you can call it nucleosome models. Whatever you are going to see, these are the different nucleosomes. So this is called as one kind of packaging of the DNA. So DNA supercoiling is a process which makes DNA fit in this cell's nucleus, and normally it regulates the gene expression. Expression means the protein synthesis. The function of DNA is it is going to give you the protein, otherwise it does not have any other functions. So DNA gives you protein, and that proteins normally regulates our characteristic, are the functions, different kind of properties. So that is called as transcription regulations. So how this DNA is going to be packaged? So we are going to start from this one. So this is roughly are the double stranded DNA. So first, this double stranded DNA, which will be in the 2 nanometer scales, means the distance from the diameter is roughly 20 angstrom or 2 nanometer. So this DNA will be wrapped on the specific proteins, that is the basic protein which we call histones. So that this structure is called as chromatin bead on a string, or you can call it nucleosome model. So this is the DNA in the form of thread-like structure. It will be wrapped on the nucleosomes. So this diameter of the nucleosome histone protein is roughly 11 nanometer scales. Then this nucleosomes will be wrapped around the hypothetical axis, and it will be going to form a chromatin fiber of the packed nucleosomes. So that will be 30 nanometer structures. Then this is going to form a section of chromosomes which will be 300 nanometer scales. Then it will forming a condensed zigzag forms, that is the condensed form of chromatins, you can call it chromosomes, which is 700 nanometer scales, and then ultimately it is going to give us chromosome. So many different folds of packaging is there in the case of our cells. So the 5 important functions why this supercoiling is required, and what is the basic significance of this supercoiling in the cells. So first is this is allowing the DNA, whatever DNA we are having long, to means that 6 feet long DNA, that is going to fit into the micro scales cells. Second function is it provides the stability to the cells. Third important function is it is going to prevent the unnecessary enzymatic attack on the DNA. So DNA will be more stable, DNA will not be digested by any exonuclease or endonucleases. Then it also regulates the gene expression, and lastly it is going to form chromatins, chromatids and chromosomes, which just now we have seen it. So one by one we are going to see it. First, it is going to allow DNA to fit into the cells. So as informed, the normal DNA length will be roughly 2 meter long, more than 6 feet long. So it means it should be highly organized so that it can be fit into the nucleus or inside the cells. So that allowing means this DNA condensation, or you can call it DNA will be so packed so that it will be accommodated into the cells, that is called as DNA supercoiling. So this DNA supercoiling allow the entire genome to shrink and settle into the cell's nucleus. Supercoiling of DNA, which is the coiling of single stranded DNA with one another, and supercoiling of duplex DNA, where one another facilitate the DNA packaging. So either single stranded DNA is supercoiled with the single stranded DNA, or double stranded DNA will be supercoiled with double stranded DNA. So both type of packaging we are having. Technically, the interaction of DNA with certain proteins force it to the supercoil by interactions, and most important protein by which it is going to associate, that protein is called as histone protein, which is the basic proteins. But as highly supercoiled DNA reduces the rate of replication, because DNA will be condensed, DNA will be very packed, so enzymes will not be accessible. So some of the enzymes like topoisomerase and gyrase regulates this process of unwinding. Whenever DNA will be compact, it will not be used for replication, transcription or translation. Whenever cells require that DNA should go for replication or transcription, that time this topoisomerase and gyrase helps them. Particularly, various histones are one among of the class of those proteins which interact with this DNA and allowing the supercoiling of the DNA. Just now I have told you, they forms the nucleosome. The second functionality is going to provide the stability of DNA. So the stability of DNA is very important for a cell to perform their metabolic activity, because maximum or almost all the functions of a cells depends on their DNA, because DNA gives the protein and protein is the functional unit of our body. So single stranded or unwinded, unpacked DNA, loosely packed DNA, they are normally unstable and they can interact wrongly. They can interact with number of components, they can be easily degraded by the different enzymes. So unstable DNA are disrupted by the process of DNA packaging in eukaryotes too. Supercoiled DNA are very stable because it is condensed, so enzymes cannot interact with the DNA. So the correct base pairing, the helix of DNA stabilizes, and through supercoiling double strand DNA, the entire genome becomes stable. So when we are having DNA that will be stabilized by base pair adenine to thymine, cytosine to guanine base pairing, and double stranded DNA supercoiling normally stabilizes the complete genome. However, to allow various enzymatic reaction, it becomes unstable, but the stability of DNA is important to form the structure like chromosomes. The process of stabilizing and destabilizing of DNA by supercoiling and unwinding controls is the process of gene expression and regulations. I have already made a video on euchromatin and heterochromatin. So whenever we want DNA to be supercoiled, or it will be very tightly packed, that time DNA are normally inactive. You can call it transcriptionally inactive. It will not go for transcription or regulation or replications. But whenever DNA will be unwinded, unpacked form, loosely packed form, that time it is going for gene expression and protein synthesis and replication also. So the packaging of DNA or supercoiling normally makes DNA stable, so that it will not be accessible for different enzymes. Then it also prevents the unnecessary enzymatic reactions, because relaxed DNA are susceptible for more enzymatic reactions, because it will be very easily accessible for them. Enzymes involved in replication, transcription and translations uses regulatory element to settle or DNA for catalytic activity. DNA supercoiling thereby does not allow enzymes to work by blocking regulatory regions and only allow when needed. This process is automatically controlled and highly tissue specific. So normally this DNA packaging supercoiling is regulated by many different functions, like different kind of chemical modification of histones, DNA modifications, acetylations, acylations, methylation, different type of processes are there. So those processes normally regulates the DNA packaging. It is also going to regulate the gene expressions. Just now I have told you normally DNA exists in two different forms, heterochromatin and euchromatin. So it is one is loosely packed DNA, other one is tightly packed DNA, very compact DNA. When DNA will be very tightly packed, it will not be accessible for different enzymes, so it will be transcriptionally inactive. So it will be not active DNA. And when DNA will be loosely packed, it will be accessible for enzymes, then it is going to for expressions. So that is it is going to regulate their gene expression will be regulated by different kind of reactions. Either histones is going to involve or different other components are going to involve in this process of gene expressions. So heterochromatin regions are the DNA which is highly tightly packed, supercoiled, and it is loosely packed, that is euchromatin. Means heterochromatin is packed DNA, highly packed, supercoiled, so it is transcriptionally inactive, and euchromatins are loosely packed, so they are transcriptionally active DNA. And lastly, it is going to form chromatins, chromatids, and chromosomes. So we have just now seen there are different histone proteins. Roughly we are having 5 different type of histone proteins. They are H1, H2, H2b, H3, H4. So these last 4, H2a, H2b, H3, H4, 2 in number, are going to form a octamer core. On that one DNA will be wrapped roughly 1.67 folds. It will be roughly around 200 base pairs, and this H1 is work as a locker, so it locks, knots the DNA on the DNA. So they are going to form say chromatin and nucleosome models. So this chromosome is a final product of DNA packaging by supercoiling, having 2 chromatids, arms and centromeres. 23 pair of chromosomes is a typical cell has, except germ cells where only 23 chromosomes will be there, only haploid cells of chromosomes will be there. So the structure chromosomes eventually helps DNA to fit into the cells and inheritance of gene from one to another cell. So these are the 5 different functions which is supported by DNA supercoiling or packaging. So this is all about for today. This is how it is going to be packed. So it starts with the DNA, then nucleosome models. This is the histone proteins on which DNA will be wrapped. Then it is B down strings, then 13 nanometer structures, solenoid structures, then chromosomes, chromatids, and ultimately metaphase chromosomes. So this is all about for today. If you have any query, any comments, you can write in the comment box. If you are not subscribed my channel, you can subscribe it. Thank you very much. Have a nice day.